Are you? I guess I can get started. Do I hear something? Okay. No, that's fine. Okay.
Maybe we can proceed with this. With me.
Okay, thanks. Thanks, Lisa. Hello, everyone. I hope you can hear me well. So if you have any issues with the audio, just let us know. I'll try to solve it real time. So today we're going to cover one of the works from our group, RawHash. So the purpose, probably we covered it last week as well, but the purpose of these presentations is actually for the seminar students to get to know like how we are doing the paper analysis, right? As also discussed in the first lecture, starting with the paper description, the details of the paper, and then the analysis of the paper. So I'll try to also mock, let's say, a student presenting a RawHash paper, although this is basically one of our papers. And I'll try to basically show like how we approach such paper reviews. So you already know, I guess, like which parts should take how much time, right? The paper presentation, I guess, around 25 minutes, and the analysis around that. Although our discussion and the analysis part probably are not going to be as interactive as it would happen in class with your peers. So don't take that part at least as a reference point, but you can take the general structure as a reference point, let's say, for paper presentations. So then let me get started. Welcome again, everyone. Today we're going to present the paper entitled RawHash, enabling fast and accurate real-time analysis of raw nanopore signals for large genomes. This paper was presented at ISMB ECCB conference last year in 2023 and published in the Bioinformatics journal as part of the proceedings. So let's start with the background, as we should always start to give some, let's say, sufficient info or background so that you can understand the rest of the paper. So let's give some motivation about why we're doing sequence analysis or genome analysis. So it is very important for many reasons. For example, it is important for understanding the genetic variations, species evolution. It is very important for predicting the presence of pathogens, let's say, in an environment. These can be harmful. For example, in the case of the several disease outbreaks, the sequence analysis can be very useful, and also, of course, it is very important for personalized medicine. And so how do we do sequence analysis? So we have these machines called high-throughput sequencing machines, which takes biological molecule, nucleic acid molecules, for example, DNA as input, and then as output it generates sequences from DNA or fragments from DNA that we call reads. And since these reads are fragmented and we don't know the origin of these reads in the genome, the goal is usually to basically have some understanding of where these fragments are coming from and how they differ, let's say, what sort of mutations they may have and so on. And to achieve these, the sequence comparison is basically very essential to understand these features, so that we can analyze these sequences accurately and quickly. And we do this usually by comparing these fragments, right? So these can be referred as the pieces from DNA, let's say, can be referred as, let's say, pieces from a puzzle. And we could solve this puzzle by either, let's say, comparing these pieces to each other, right? To maybe be able to understand what these pieces of puzzles look like. Or we might have a template sequence that we call reference genome, and then we can compare these pieces or DNA fragments to that reference genome to essentially map these pieces to their corresponding regions in the reference genome. And this is very important. Basically, it's very essential to understand the functionality of a sequence, mutations and diseases, and so on. And this is one example, one practical example, how we map reads to our reference genome. So in this case, we have a reference genome. This can be huge, right? In the case of human genome, we have around 3 billion characters, and we have a fragment of DNA which we call read from the high-throughput sequencing machine. And there are mainly 4 steps to map the read to a reference genome in a practical manner. The first step is done only once, usually once per reference genome, which is basically constructing a hash table for a reference genome that contains certain subsequences or fixed length subsequences that we call k-mers, as well as their positions where these fixed length subsequences appear in the reference genome. So we call these subsequences k-mers, and then we extract those k-mers from reference genome, and then we basically store these k-mers as keys in the hash table, and as values we store the positions where they appear in the reference genome. And similarly, when we have a read that we want to map to a reference genome, what we do, we similarly extract k-mers from read, and then this time we query this hash table to identify where these k-mers appear in the reference genome very quickly. So this is simply a single lookup of a hash table. And then when we have these matches that we call seed matches, some of these matches can be, let's say, incorrect. So you want to minimize the number of false matches of these k-mers in the reference genome so that the further steps, the next steps in read mapping are relatively cheaper. So for this, the approaches called seed filtering are applied, or chaining, to prune basically uninformative or unreliable seeds. And then comes the alignment part where it tells you the exact differences between a read and its corresponding position in the reference genome. So this is, and basically the accuracy and the speed of read mapping is also mainly affected by the output that you get from a sequencing machine, because each sequencing machine generates relatively different types of output. One technology known as Illumina sequencing is essentially known to produce very short pieces, while those pieces, fragments, are relatively accurate. The other types of technologies known as PacBio or Nanopore sequencing, they usually produce longer reads, which makes read mapping relatively easier now that we have longer pieces, but they are usually more erroneous than the fragments that you get from Illumina. So these are basically the 3 main types of sequencing technologies that we have, and each of them has, let's say, they generate different type of raw data, which are usually then translated into the basis of DNA characters of ATGCs, right? And today, we're mainly going to focus on this particular technology called Nanopore, due to its unique benefits that we're going to explain now. So Nanopore sequencing is a widely used sequencing technology, so it can produce fragments of up to like 2 million characters, right, which is very important. And it also provides unique features such as portable sequencing. So you can just fit it in your phone, and then this is such a small device, and then you can do your sequencing in a portable manner. And it is also relatively cost effective. You can sequence a human genome maybe 3 times by only paying around $1,000 US. And there are also some unique features such as real-time analysis enabled by this sequencing technology. So let's see what a real-time analysis is and how Nanopore sequencing works. So here, what we see is essentially on the left you have it. You can assume 3-layer solution. The top layer, the middle layer, a membrane, and the top layer is negatively charged. There is a middle layer membrane, and the bottom layer is positively charged. And what's happening here is that in the membrane we have a tiny pore that we call nanopore, and on top of it there is a mother protein. And what happens is that since DNA is negatively charged and it's initially placed in the negatively charged area in this 3-layer solution, what's happening is that it's literally moving from negatively charged region to a positively charged region. And as it moves from this side to this side, it actually goes through this particular pore, which actually then measures the ionic current disruptions, ionic currents that are disrupted as this particular DNA moves through that tiny pore. And these ionic current measurements are generated as DNA moves through the pore at a certain speed. It has a particular throughput. which is around 450 nucleic bases per second. And then what we can do, as these raw electrical signals are generated by our sequencing the DNA, we can actually perform analysis or even real-time analysis if your analysis speed can match the data generation speed in nanopore sequencing. And the analysis can either be perhaps you translate these signals into ATGCs quickly, and then you do your usual analysis, like in read mapping that we showed earlier, or perhaps you can even don't translate these electrical signals, but still perform analysis. But in either case, what you can do is that based on your analysis, you can tell the sequencer, for example, to stop sequencing a read at any point if sequencing that read is no longer needed for us, maybe it is unnecessary. And this is basically pretty useful for two reasons. So these are basically, these can only be enabled, especially this decision making can be enabled effectively with real-time analysis. So then let's cover the benefits of real-time analysis, right? So one benefit is, usually what we have is that we first sequence the DNA, and then after sequencing, we analyze it. But real-time analysis enables us reducing the latency by overlapping the sequencing and the analysis time, right? And the second, the most common approach is to, of course, to completely sequence a read and then analyze it. But what we can also do with real-time decisions, we can partially sequence the read, which can then translate into reduced sequencing time and cost, because we don't unnecessarily sequence reads if they are not useful for us. And there are certain challenges to achieve real-time analysis. For example, the first challenge, the most important one, is to achieve rapid analysis, right? We want to match the throughput of data generation. Second is we want to make decisions as quickly as possible, because we don't want to sequence unnecessarily. And of course, we want to analyze this noisy electrical signal data accurately so that our analysis and our decisions are accurate. And also, of course, for portable reasons, for scalability reasons, we want to make all these computations power efficient. And one more, perhaps, piece of information before we dive into the paper, RawHash, which is basically about how these raw electrical signals are essentially enabled, are analyzed, and then and compared against a certain database, a reference genome, to find similarities. So in Nanopore, as we explained earlier, we have this particular membrane here, and in the middle we have this nanopore, right? And depending on the chemistry that you're using, depending on the nanopore model that you're using, there is a certain amount of nucleotides, bases, at a time inside the core. And this number is around 6 to 9, depending on the model that you're using. So what we know that there are K-mers or K-nucleosides inside the core. So you can assume that maybe we have this particular K-mer at this moment while this DNA is being sequenced. And the other terminology that we want to introduce is an event. Event is basically nothing but a particular segment in the raw signal. And this segment is assumed to correspond to a particular K-mer, sequencing of a particular K-mer at a certain period of time. And in this case, if we basically label this region of the signal as an event, and this perhaps may mean that this event is corresponding to sequencing of that particular K-mer at a certain point. And usually, since DNA is moving through the pore, and these bases are of course shifting as it moves, there are abrupt signal changes in the signal as this DNA moves through that pore, which indicates us there is a new base coming into a pore and a base coming out from the pore, so that we can identify the next K-mer in the signal. And statistical methods are usually used to find these abrupt changes so that we can identify all possible K-mers in the signal. And then what we do is that we simply, rather than labeling this region with a K-mer, we simply label this region with a signal value, which is basically the average of signals inside that region of the signal. And this helps us to essentially find similarities between raw signal and a reference genome, as I'll explain next. But there is one key observation that you should keep in mind is, which is basically, we observe that the identical K-mers generates similar event values or similar signals during sequencing. So this is one key observation that is very helpful for RawHash data. Go ahead.
So the entire genome, so like the 6 letters in this example, could be derived from just this one value?
Almost, yes. There are like some normalization steps and some, let's say, outlier detection steps, but you can make almost a pretty good guess what sort of k-mer that you might have there, depending on this value. Although it's not always like 100% accurate.
Yeah.
It's accurate enough.
I was just trying to answer like in principle.
Yeah. So yeah, then let's start covering RawHash then. We'll start with an executive summary. So the problem that RawHash tackles is the real-time analysis of raw Nanopore signals is inaccurate or inefficient, especially for large genomes. And the goal of this work is to enable fast and accurate analysis of raw Nanopore signals for large genomes. And there are 2 key contributions in RawHash. The first one is it provides the first hash-based mechanism that can quickly and accurately analyze raw Nanopore signals. And the second contribution is called sequence until that can accurately and dynamically stop the entire sequencing run, not stopping the sequencing of a single read, but the entire run of sequencing, if the computation decides that the further sequencing is not necessary. And based on the extensive evaluations across 3 use cases and 5 genomes of varying sizes, RawHash provides significant improvements in performance and also substantial improvements in accuracy. And also RawHash shows that the sequence until mechanism can reduce the sequencing time and cost by 15x by enabling stopping the entire sequencing run completely. So let's cover the problem and also the prior works. So there are usually 2 types of existing solutions to achieve real-time analysis. One is to use, the most classical method is to use deep neural networks to translate these raw signals into the basis of DNA characters, ATGCs, and then this means that you can do all your read mapping, all types of classical analysis later on, as long as your analysis is fast enough. So the advantage of this approach is that this provides less noisy analysis, meaning more accurate analysis from base code sequences, but this is usually very costly and power hungry. It requires costly and power hungry computational approaches. The second direction is mapping the raw signals directly to the reference genomes without translating them into ATGCs. So here we're mapping the electrical signals to reference genomes. And the 2 main advantages of these approaches are first, raw signals contain richer information than just bases. So it's known to contain essentially extra information, whereas you would only have ATGC information in like the base code your signal. You're losing some information basically. And second benefit is that it enables efficient analysis with better scalability and portability, because these computational methods usually require less, let's say, computationally complex approaches to map that read to a reference genome without using deep neural networks. But there are still like certain problems related to mapping raw signals to reference genomes. So assume that you have this particular raw signal, and then you might be mapping the signal to any type of genome, right? But let's take 2 types of genome. One is a small genome, the other one is a large genome, which is maybe, for example, a human genome. So when you're mapping your signal to a reference genome, this means that you have fewer regions that you want to focus, that you can focus per read at a time, right? This means that since there are fewer regions to focus, maybe then you can make more accurate decisions, right? You don't have to confuse your computation with too many regions. And the second is, since again you're processing fewer information, then this means that you can process it faster per read at a time. So this means that you can achieve relatively high throughput with small genomes. But as you move to larger genomes, so this means that you start having substantial larger number of regions to check per read, to analyze per read, as the genome size increases. And this is very problematic because as you have many basically regions to check in the genome, this can confuse your computational mechanism, especially if your computational mechanism is based on some probabilistic decisions, probabilistic approaches. So this can lead to inaccurate mapping. Or if you are performing some costly operations per read, per region to check in the reference genome, and as the number of regions increases per read, then this means that you're doing a lot costly operations. If you're already doing costly operations per read, for example, distance calculation, then this means that your throughput is getting reduced as the genome size increases. And this may lead to also performance problems. And what we find is that existing solutions are either inaccurate or slow for large genomes. And the goal in RawHash is to enable fast and accurate real-time analysis of raw nanopore signals for large genomes. And to this end, RawHash has two key contributions. One is the first hash-based search mechanism to quickly and accurately find similarities between a raw signal and a reference genome. And the second contribution is sequence sample that can dynamically stop the entire sequencing run if further sequencing is unnecessary. So we will cover the first solution now, first contribution now. So again, remember the key observation that we also covered earlier, which is the identical nucleotides or identical K-merges generate similar raw signals. So this means that they don't generate exactly the same raw signal, but they generate similar raw signals. And to exploit this observation, an earlier work actually performs distance calculation between a pair of raw signals. So if the distance, for example, an Euclidean distance between a pair of raw signals is small, then this can indicate similarity between them. However, as the genome size increases, given that this distance calculation is very costly, this becomes impractical for larger genomes, and we cannot do real-time analysis. And instead, in RawHash, what we want to do is that instead of doing distance calculation, we want to somehow generate a hash value from raw signals so that we can enable fast matching rather than doing distance calculation. But of course, there are certain challenges to it. The first challenge is these raw signals are not exactly matching, right? And the challenge is how can we generate the same hash value from similar enough signals, right? And the second challenge is we want to accurately find as few similar regions as possible. So it's not like we want to find millions of matching regions per read, even though your genome size is, let's say, in the order of, again, millions of bases, right? So we want to really find as unique hash matches as possible between a read, between a raw signal and the reference genome. And this is the overview of RawHash. So how it starts is first, it converts the reference genome and the signal to its events, right? So remember, we covered the notion of events. So we can convert both reference genome and the raw signals into series of event values. These are somehow averaged signal values. And then to essentially alleviate this variation effect, meaning that the signals are similar to each other, but not exactly matching if these cameras are the same that are being sequenced. So to elevate this issue, it quantizes the signal values into, let's say, smaller set of values, or it buckets. It's called a bucketing approach. And then based on these quantized values, now we can perform some hash matches because now there is no variation effect, hopefully. And then based on these hash matches, we find some matching regions, and then we perform chaining and mapping to identify the similarities between a raw signal and the reference genome. So the steps on the left are called indexing, and this is just done once per reference genome, and this can be done offline not in real time. But the steps on the right are real time because your raw signals, your signals coming from a sequencer are generated also in real time. So we need to be able to do all these steps on the right in real time again. So let's cover each of these steps one by one. So let's start with reference to event conversion, right? So how do we convert the reference genome to some sort of signal values, right? So for that, what we do is we start with extracting the k-mers from a reference genome, all overlapping k-mers from a reference genome. And the size of this k-mer is actually crucial because we determine the size based on the nanopore model that we are using at the moment, because we know on average how many chimeras that will exist inside a nanopore at a time. And of course, and if basically this number is 6, we extract 6 mers from a reference genome. And what we do is that we have this lookup table that gives us the expected signal values. So these are essentially based on some statistical analysis and so on. So given a chimer, it will give you what is the expected signal, expected event value for that particular chimer. So using that, you can simply convert each chimer to its expected signal value, and then you can do some normalization and so on later on, so that you're able to match these with the signals that are being sequenced in real time. So this is basically how we convert the reference genome to its synthetic signal representation. So similarly, we also need to be able to convert the signal, raw signal, also to events, right? So events meaning potential chimers that are being sequenced at a time. And what we do is that we simply use a statistical test known as a segmentation or t-test to spot these abrupt changes in the signal. And these abrupt changes are then identified, and then we calculate the means between each abrupt change, and then we assign the event value accordingly, and then similarly do normalization and so on. And now we also have a series of events and series of event values for raw signals for coming from sequence also. So this means that we have now consecutive events, meaning perhaps we have now consecutive chimers similar to the reference genome. But the question is, can we now directly match these signals to each other so that we can identify the matches? So to explain this, I'll cover the quantization mechanism. So remember, there's slight differences in raw signals coming from identical chimers, right? Meaning, if you have this particular chimer, if you are sequencing it at different times, so this may so happen that after normalization, you may get different normalized event values, right? So you may have this value, and at another point you may have this value, and so on. So these values are not necessarily exactly matching, but it is expected that these normalized values will be close to each other. And given this observation, one challenge is that we cannot simply match these normalized values to each other, because there will be variation. So we need to somehow remove this variation effect. And the key idea to do so is to quantize these event values. So this means that we enable assigning the identical quantized values to similar event values, right? So this is like bucketing from a large set of values to a small set of values, such that similar close by values will get assigned to the same bucket, essentially. And so then this is the quantization mechanism. So then let's cover the hashing part. So there's one more challenge you should handle after quantization. So each event, right, that we're quantizing usually represents a very small chimer, which is around 6 to 9 characters. And if you were to simply just try to find a match of such a small chimer in a human genome, you would simply find perhaps millions of matches in the genome, because it's likely that you're going to, the human reference genome will contain this particular chimer a lot of times. Although the read that we're sequencing at a time may not necessarily be coming from the region that we're finding these seed matches. So the question is, how can we make this longer? How can we make this information longer? So to achieve this, what we do is that we create longer chimers somehow from consecutive events. And to do so, we simply pack the quantized values into a single value while preserving their order in our packing, so that we sort of create a longer chimer from many consecutive event values, and then we simply hash this, and then we use this hash value for matching purposes. And this is basically the entire picture of generating hash value and then using the hash table to do so. So again, to cover almost all of these steps, the left side is the offline step, the right side is the real-time step, and we already covered how we generate hash values from reference genome and the signals right? So the next step is simply in the offline step, when we generate hash values, we store these hash values in the hash table. And then this is the end of offline step, right? And then later, what we do is that in real time, as the reads are being sequenced, and as we generate the hash values from these raw signals, what we do is that in real time, we query this hash table. And then if there are some matching regions, matching hash values between a read and this particular hash table or the reference genome, what we do is that we get these matching positions and then we do some chaining, which essentially finds some collinear, let's say, ordering of these matches of hash values to indicate that there could be really a similar region between a raw signal and the reference genome. And based on this information that we generate in real time, we continuously ask the question, should we continue mapping or not? And if the answer is yes, we simply retrieve the next chunk of data that is generated in real time and then do all of these steps again. Or if the answer is no, we stop sequencing that read by activating features of Nanopore sequencing that are called read until or run until. And by doing so, we can simply stop sequencing a read or the entire sequencing run and move on to the next read. So this is the first contribution of RawHash. So I'll very quickly cover the second contribution in RawHash, which is the sequence until mechanism. Slightly, basically a different approach, but they somehow nicely merge together. So the problem that we're covering in sequence until is unnecessary sequencing, of course, wastes time, money, power, and so on, and we want to avoid this, right? What we want to do is that we want to dynamically decide while sequencing if further sequencing of the entire sample, the entire DNA sample, is necessary to achieve high accuracy. And if the answer is no, if we don't need to sequence further to achieve high accuracy, we can stop sequencing early without sacrificing accuracy. The potential benefits of this approach is it can provide significant reduction in sequencing time and cost, and basically we evaluate this. We evaluate the benefits of sequence until based on a particular use case that is known as relative abundance estimation, where you try to identify the ratio of each genome in your sample in your analysis. So how does the sequence until work? There are a few key steps. First, what we do is that we continuously generate relative abundance estimations after sequencing of n reads, right? So, for example, you sequenced, I don't know, 100 reads. Now, what you can do is that you can perform some estimation of the organisms that are available in your sample. Then you generate some estimation, right? It's called estimation number one. So what we do is that we keep doing these estimations, and then we keep the last t estimation results basically as we generate these estimations, right? We have this history of estimations now. And then what we do is that we try to detect an outlier in these estimation results by performing cross-correlation of the recent t results, t estimations. So we do this outlier detection because if basically there is no outlier in any of these estimations, then this means that our estimation did not actually change much. Although we sequenced more data, more read, our estimation kept almost constant, kept almost the same, right? So there is no outlier. So this indicates that even if I continue sequencing the reads, even if I continue sequencing my sample, it's likely that my estimation won't change, right? That's the key idea. Because we observed that in the last t estimations, maybe in the last 50 estimations, it didn't change, and it's likely that it won't change also in the future. So then we can make the decision to stop sequencing because it is further sequencing is likely to generate a consistent result, and then we can stop sequencing. But if there's an outlier, meaning we basically see that there are substantial changes in the estimation, and this tells us that further sequencing is useful because it's still changing our estimation substantially. So we want to keep sequencing further as long as we see an outlier in our estimation. So then this is basically providing us some benefits to stop entire sequencing run by dynamically deciding if it is necessary or not. So this is basically two contributions in RawHash, and I'll quickly cover the evaluation part in RawHash. So RawHash is compared to two state-of-the-art works, Unquote and Sigma. There is a high-end CPU baseline, and each tool uses 32 threads. There are 3 use cases that are used in comparisons. One, read mapping reads to reference genome. Second, relevance estimation. The third, contamination analysis, which aims to find whether a sample is contaminated by a certain organism, for example, a COVID genome or not. There are 3 evaluation metrics. One, throughput, which is basically the number of bases each tool can process per second. The second metric is potential reduction in sequencing time and cost by activating read until or run until. And the third is accuracy of mapping information. And for that, we use Minimap2 as a baseline and mapping output, which is generated from base code sequences. And then we measure precision recall and advanced scores. And then we use various data sets from very small genome, a viral genome of COVID, up to a human genome of size 3 billion bases. And we also have relative advanced estimation, which is a mock community constructed by combining all of these data sets, and the contamination analysis contains a sample from a COVID and a human genome. So the first result is about throughput. So here the figure shows on the y-axis the throughput results of each tool, and the x-axis shows the data sets used to generate these particular results. And we have 3 tools that are indicated with different colors here. And one thing to remember is that real-time analysis requires faster throughput than a sequencer, right? And the throughut of a sequencer is around, throughput of a single nanopore is around 450 bases per second. And basically, this 450 is around here. And if a bar is above that threshold, you can assume that the tool can achieve real-time analysis. And if it is below that threshold, it cannot achieve real-time analysis. And the first result that RawHash shows is it provides significant improvements of around 26x and 3.5x better average throughput compared to uncalled and sigma, respectively. And also the second observation is for larger genomes, for sigma, it cannot perform real-time analysis due to the costly distance calculation that we discussed earlier. So the second result that we show is the potential benefits in reducing the sequencing time and cost. And here on the y-axis of this figure, we show the average number of bases that each tool needs to sequence before making a mapping decision. And here it is lower is better, because it means that you need to sequence less. And then again, on the x-axis, we have the different data sets. And we only compare RawHash and uncalled due to their good results in the previous metric. And in this case, what we observe is that for larger genomes, RawHash reduces the sequencing time and cost up to 1.3x, because it requires less bases to sequence for these larger genomes, and for smaller genomes, uncalled seems to be better. And let's look at the accuracy as well. Here we show the mapping accuracy of each tool for all data sets. And what we see is that, again, for large genomes, RawHash significantly improves the accuracy of read mapping, almost up to 2x here in F1 score, which is a combination of precision and recall, both for basically human genome mapping and also relative estimation. And for smaller genomes, although RawHash seems to be slightly less accurate, the accuracy results are relatively close to these tools. And we also look at the relative estimation results. We see that RawHash provides the best estimation result. And also the second result that we look at by looking at the relative estimations is basically the benefits of sequence sampling mechanism when it is integrated in RawHash. And what we do here is that we use the entire read set with RawHash and then perform some estimation, and then we activate sequence sampling mechanism with RawHash and then let it decide when to stop sequencing. This means that it will only use a portion of the entire data set to make an estimation, relative estimation. And then what we see is that sequence samples stop sequencing after sequencing around 7% of the entire data set, entire sample dynamically. And then it can still provide relevance estimation that is very close to the estimation that RawHash makes by using the entire data set. So here we have the use of 100% versus 7%. This correlates to 15x, let's say, reduction in the sequencing time and cost, while it can still achieve almost identical results that the RawHash generates with the entire data set. And we also simulate the behavior of sequence sample in the other tool, uncalled, and then see what happens. if we also integrate sequence sample with that, and then based on our simulated results, we also see that sequence sample can actually significantly improve the sequencing time up to 100x with these tools. So to conclude, there are two key contributions in RawHash. One is first hash-based search mechanism, the second is sequence sample mechanism. And it improves the throughput, accuracy, and sequencing and time and cost significantly by its efficient and accurate mechanisms. And based on this paper, the authors conclude that there are many opportunities for analyzing raw signals in real time. For example, using existing solutions for hash-based search and using also cheap indexing and so on. So this is RawHash, and with that, I'll quickly go into the analysis part of this paper. So there are several strengths of this paper, and the first strength is it provides the first mechanism that can accurately analyze large genomes in real time, and because it provides the best accuracy and performance, the average accuracy and performance for large genomes. So the second strength of this paper is, I believe, the sequence sample mechanism, which is a new technique that can dynamically, the first technique that can dynamically stop the entire sequencing run based on the real-time analysis, right? It takes a dynamic decision to stop the entire sequencing run, and it is important because it has the potential to substantially reduce sequencing time and cost. The other strength that I see in this paper is it provides the first hash-based search mechanism for raw electrical signal analysis, and achieving this is enabled by the novel quantization mechanism. And so this also tells us that there are many opportunities in this first hash-based search mechanism. With the first hash-based search mechanism, it is clear that there are many opportunities to utilize the existing hash-based search strategies that are used for base call sequences. Also, we can use them for raw sequences, perhaps. So these are the main strengths in RawHash. So then, what are the weaknesses? So we see that RawHash is usually less accurate for smaller genomes than other tools. Also, what we see is that the throughput decreases as the genome size increases, right? So this means that there is a certain limit on the genome size that RawHash can provide the real-time analysis, right? It cannot only go up to a certain amount for real-time analysis. And also, what we see is that RawHash needs more bases to be sequenced per read for smaller genomes. So this means that potentially increases sequencing time and cost for these small genomes. And the other weakness is that misses the signal alignment functionality, which can be a key functionality for particular applications to achieve better sensitivity. So these are the weaknesses. So then let's go into the discussion points and ideas that we can use in this paper. So one discussion point, or perhaps an idea, maybe it could start by asking a question, which is, what are the existing, if you have any knowledge on this, what are the existing hash-based mechanisms that are used for base call sequences and can also be exploited for raw signal analysis, right? Here you can ask like similar questions to your colleagues to engage a discussion and so on, so that they can answer. But here I won't wait for answers, so I'll just reveal. So one particular answer is the sketching techniques for a sampling database, right? So there are certain sampling approaches that simply do not take all k-mers, but a certain subset of k-mers, so that you can reduce your database sizes and still improve the performance due to the reduced database size. And this is basically can be imagined by placing the sketching technique here right after hashing. As soon as you have your hash value, then you can perform your sketching or sampling, maybe for example using minimizers, droppers, and so on. And then based on that sketching output, and you can store the resulting hash value to the hash table, and this means that you can further sample your the hash values that you generate from raw signals and the reference. This is the sketching technique. We can also use the existing fuzzy seed matching techniques right? We discussed in an earlier lecture, which enables mismatches between hash value matches, right? Even so, usually what happens is that when you generate a hash value from a string, let's say a value, it is expected that if the hash function that you're using is a local hash function, then if your hash value is matching, then the input to that hash function is also probably exactly matching unless you have a collision. But then the question is, can you actually also generate hash values although the inputs are different? And here we address this. These authors address these challenge to enable fuzzy state matching, and then this can be used also for raw signal analysis as well. So how can we make RawHash also more accurate, right? Maybe we could do this by enabling more accurate quantization. So what are the other quantization approaches other than the ones that are used in RawHash? Maybe we can do some more accurate filtering of seed matches. Maybe we can reduce the number of seed matches or accurate chaining. And also, how can we make the chaining itself, chaining algorithm itself more accurate as well? And it so happens that these improvements are also described in this particular paper. So you can go ahead and read how these are implemented to achieve more accurate results. And also, the other question is based on the weaknesses that we had. How can we further reduce the average number of bases to sequence, right? And then it turns out that when you achieve more accurate mapping, this also directly translates to reducing the number of bases that you need to sequence, because you can now be more confident about your decisions, and this means that you can make early decisions to stop the sequencing just because your mapping is more accurate. And the next question is, can we add read alignment to RawHash, and would adding the read alignment make the RawHash slower because it's an additional step, additional computational approach? And turns out this is also explored in a paper, RawAlign. And since also we can make the mapping decision more accurately, you can also optimize other parameters so that you can still be accurate and still faster, relying on the alignment information that the RawAlign generates. And the last question, which is in my opinion still unanswered, unsolved, which is, can we make the throughput constant even though genome size increases? So this is still a question that bothers me, and if this can be also achieved, I think that will be a huge contribution. So with that, I'll conclude my RawHash presentation, and yeah, I don't know if you have any questions I can answer. Now we can switch to RawAlign. Do we have any questions?
Wait, maybe one question. There's this outlier criterion for solving this concern.
Could you say that?
The outlier criterion. I didn't really understand how that works, because the solving criterion is that you sequence if there is an outlier in the set of samples,
in the set of estimations, yes.
You continue with this, if there still is an outlier.
Still there is a?
If there still is an outlier, you continue with the sequence.
So what happens is, so assume you iteratively generate some estimation. Like assume you have 3 genomes in your organism, maybe bacteria, viral, and human genome. And let's say you sequenced 100 reads, and then maybe it's time for you, after sequencing 100 reads, to make some estimation based on the mapping information, right? Because each read maps somewhere. And based on your estimation, you see that 30 of these reads map to bacteria, 40 of them maps to viral genome, and 30 of them maps to the human genome, right? Now your estimation is like 30%, 40%, and 30% relative for each genome. And then you sequence next set of 100 reads, and then now you have 200 reads, and then you make your estimation again. And then your ratios perhaps change or don't change. And then you, now like you had your first estimation, which is 30, 40, 30. And second estimation is something, and so on. Now, perhaps let's say you have 50 estimations, the history of estimations. So what you can do, you can actually consider them as vector values in a vector, each estimation, right? What you can do is that you can perform cross correlation by simply multiplying each of these vectors. And if there is a particular vector that generates substantially large resulting value when multiplied to all other vectors, this signals that essentially this particular vector is an outlier in the history of your estimations. And if there is an outlier, it actually indicates that my estimation changed substantially. Why did it change? Because I sequenced that a certain amount of data, maybe 100 reads, and that particular 100 reads changed my estimation substantially. So this is like, then I'm like, oh, then maybe I should keep sequencing it. I should keep sequencing further until my estimations start converging to a particular set of estimations. And then we sort of notice this convergence, right? When the history of your estimations are almost the same, very similar to each other, then we can say that, well, I kept sequencing, maybe I sequenced 1,000 reads or I don't know, 10,000 reads, but when I look at my history of estimations, my estimations still remains the same. So this tells us that even if I continue sequencing, then my estimation will stay the same. So the question is, should I keep sequencing it until the end? And maybe I'll end up just generating the same estimation that I just generated now, maybe after sequencing 7% of the entire sample, or should I stop? So we take this stopping decision because we assume that even if we keep sequencing it, the end result after sequencing fully will be very similar to what we have now, just because of that history of information that we generate. And this is mainly because we assume that the order of the reads that are being sequenced is random. Is not simply like you first start sequencing a viral genome and then there comes bacteria, there comes a human. It's like the assumption is all the reads from any organism can be sequenced at any point. So there is that randomness. So that's why we can make such dynamic decisions.
I was just wondering, so if at some point there is an outlier,
yes,
why does it stop being an outlier if you continue sequencing?
So it's essentially because in the future, you might just simply have a sufficient amount of data, right? That gives you a certain confidence that the new chunk of data that you add into there doesn't change your estimations much, right? Maybe initially it changes because it maybe moves a lot, right? Because the amount of overall data that you have is small, so there is room for like maybe different estimations. But as the total collection of data grows, even though there's a tiny part is inserted again to that collection of data, it's so basically small that perhaps it's not powerful enough to change your overall estimation.
I have one other question.
Yeah.
So we construct, I think it was called the indexing step, we construct a hash table.
Yes.
And this hash table is a mapping from the hash values to the positions of the reference.
Yes.
And I'm basically wondering if we can just, isn't it possible to pre-compute so like for, let's say we have like k equals to 6, like 6 members, then can't we just for every possible of these k-mers just pre-compute the signal value independently of this particular reference genome, and then just after this looking at this reference genome, construct the hash table which then maps the values to the positions.
That's actually what we do. So do you remember the k-mer lookup table? That's what it is. So it contains all possible k-mers. and their corresponding values in the lookup table. And if you have 6 winners, then this means that you have 2 to the 12 possibilities that you can have because of 4 to the 6 thing. Yeah, of course. If I can do it quickly here. This one. So here, is this what you're referring? We have a k-mer.
Okay.
And then this lookup table is like, tells me, it contains all values, basically values for all possible 6-mer inside it, and it's not too big because the 6-mer to the 12, and it's going to be 9-mer to the 18, still manageable, and you simply do lookup for all k-mers that you extract.
Okay, so but potentially we could do this like for literally all k-mers and not just the ones which appear in the reference genome.
True, but then you wouldn't have a position for them because you're also interested in the positions in the reference genome. So you might match, you might find that k-mer in your read, but then it becomes like, okay, I know it's let's say we know that like what sort of k-mer it is, but it doesn't appear in the reference genome, so it's not very useful for me at that point. But what you're referring to is somehow similar to earlier approaches that is called event calling. It is similar to base calling, where people try to convert signals from here to the k-mers, right? Then it's like you're somehow looking at this lookup table from the other side. Like you're somehow telling, this is my value, what would be the k-mer? But of course, like the computation of it is more sophisticated, more than just like simply looking at the k-mer. And then they did the inverse direction. They moved from signal to k-mer, and then they tried to match the k-mer from signal to the k-mers in the reference genome. So it's actually a good idea, but turns out that you need actually some hidden Markov models to accurately, let's say, classify what sort of k-mers, and even then it's not very accurate.
And is that on the other side? Is it expense? Like how expensive is it compared to the practice?
This is pretty cheap, and this is offline. Even that is like, because you do it, reference genomization to be constant, right? You just do it offline, it just stays there, and then whenever you need to use it, you already assume that these are generated. These are in your hash table. But I'm not sure if you want to touch that point. Like, what happens if we do it on the fly, I guess, even if it is not constructed earlier, should we do it on the fly while the mapping starts? Maybe there are use cases that needs on the fly hash table construction, but in the cases that we're looking at, there's no such case. We assume that the reference genome is pre-constructed. Yeah, but it is cheap. Yeah. Thanks. Good questions. Do we have more questions? Okay, and thanks everyone for your attention. And now move to the next presentation. Oh, I'm not sure.
So you can decide to go.
All right, should we continue then? So this second paper today is RawAlign, and it fits well with the previous one, so it will be a much shorter presentation because I will go over a lot of background at 500% speed. So the full title is RawAlign, accurate, fast, and scalable raw nanopore signal mapping by combining seeding and alignment. The keyword here being alignment. So you saw this background, right? We have these nanopore sequencing machines. There's other technologies, but these ones have some unique advantages. And they have also some really unique challenges in their analysis. In particular, if we want to do this real-time analysis, we're kind of looking for low latency because this is this feedback loop with these real-time decisions to improve our, say, useful throughput or good put, as networking people might say. So our decisions should be both accurate. So this feedback loop makes essentially accurate decisions. So you just don't discard useful information, and you don't keep useless information. And it should be high throughput because you want to sequence it many pores. It should be low latency and so on. If you can do that, then the overall latency of your sequencing process can be reduced, but you can also reduce both the, let's say, overall sequencing time itself and sequencing cost itself because of these real-time decisions that improve how much useful sequencing time you get out of your pores. Okay, as I said, this should be have with high throughput. It should have low latency. Latency, actually, I'll get into that a bit later again. But latency can actually be interpreted in two ways. First of all, in terms of maybe nanoseconds or microseconds, so actual wall clock time, but also in terms of how much information you need until you can make a decision. So both of them are kind of connected, but also separate metrics to evaluate. Accuracy is of course super important, and then power efficiency. Well, it's a challenge, but it's not something we specifically evaluate here. Comes kind of with performance. So the real-time analysis that we had up to this paper kind of failed to scale to large reference databases. So this could, for example, be the human genome as a reference genome. What I mean by failing to scale is it either doesn't perform well in terms of throughput. This has these baseline tools on-called SIGMAP that you just saw. Or we have RawHash, which has good throughput. It's the first work to have reasonable throughput for large genomes, but its accuracy leaves much to be desired. For these large data sets are much more difficult to decide accurately. So our goal in this work was to basically improve all the target metrics. We want high accuracy, high throughput, low latency, low memory usage, and we want a few bases to be sequenced before we can make a decision. This is the, let's say, second way to look at latency. And we want all of these things to really generalize for a wide range of reference databases. So this we propose RawAlign. This is the first seed filter aligned mapper for raw nanopore signals. In our evaluation, we observed that this is the only tool to accurately map raw nanopore signals to both large and small reference databases. It really generalizes across any data set that we tested, and it has, well, it's much more accurate than the baseline RawHash, for example. because we add some algorithmic steps that improve accuracy. But it turns out that if you optimize them well, it doesn't really cost you a whole lot of throughput. In fact, sometimes it can even improve your throughput slightly because more accurate analysis, you make more accurate decisions, and more accurate decisions can also save your computation. Yeah, right. So this is my way of showing what John showed earlier. Traditionally, you have these Nanopore sequencers. You would give these raw signals to a base caller. You would get out ACGTs and kind of run these traditional base call read mappers. For this work, this, let's say, conventional analysis pipeline actually has some special relevance. And that's because it's really kind of parallel to these raw signal analysis. So hopefully, right, because at the fundamental level, it doesn't work much different. Yes, the data type gets kind of changed by the base caller, but conceptually they do the same job, these base call read mappers or these raw signal read mappers. So hopefully we can draw some inspiration from the base call read mappers. And as it turns out, if you stretch your imagination a bit, you could take the exact same algorithms almost, just change them to the right data type. Yes, you need to deal with, that comes actually with a lot of practical implications of how these algorithms work, how they perform. You need to adapt them a bit, but in principle, conceptually, they are analogs. Okay, so, well, John showed these things, I think. The fact that small reference genomes are, let's say, relatively easy, but large reference genomes are hard. I can get back to that if there's any question about it, but let me skip straight into RawAlign. So, yeah, these were our goals. Again, all target metrics, accuracy, throughput, latency, and so on. And this was our starting point, RawHash. And you have these algorithms where you have so offline steps that convert the reference genome into a raw signal first, and then we quantize and build a hash table. And we have the real-time steps where we convert the raw signal from the Nanopore sequencer into a quantized signal and hash it, and then make lookups in the hash table and try to get out matching regions and through chaining and mapping positions. If we visualize this in a slightly different way, one could show RawHash something like this. So the x-axis, you can see positions in the read. The read is a string, for example, well, or a floating point array, I guess. And on the y-axis, you have the reference position. Let's say a good match between a read and the reference would probably be somehow along a diagonal or an end diagonal, or whatever you want to call it. The point is, when you progress one character or one nucleotide in the reference, you probably also progress one nucleotide in the read. So you advance on both axes, that's why you end up on a diagonal. An anchor is just an individual seed hit. So it kind of represents a short exact match between the two. The seeding step from RawHash kind of identifies multiple individual points in there, right? Notably, they're not connected during seeding. During seeding, you just have kind of a list of, okay, I saw the two sequences match here, there, but I really have no way to summarize them as one larger object. Then during chaining, we start connecting them, right? We try to look at all our individual hits and try to find ones that make sense together. And we group them into a chain. So clearly now they're kind of connected, but I connect them as kind of this dotted line because still a relatively loose connection. You just check the relative positions of the reads. You just say, okay, from here, it would make sense to keep going diagonally. I expect real matches to always go diagonally between these two axes. And you just check, do their positions kind of look such that this could be a feasible match, right? For example, the first, let's say the first match between the read and the reference is on the top left, then I would expect, okay, if the read, the next hit on the read is about 120 positions later, that should also be about 120 positions later in the reference. Okay, that's the case here, so this kind of looks like a reasonable connection. You can chain them together. What the chaining also does is it decides that some seed hits don't really match. For example, because it falls off the diagonal, right? The one that's grayed out, that's a real example of a read. The chaining algorithm actually decided, okay, that one seed over there, it's not nicely on the diagonal, it's probably not part of the read, and it didn't get included in the chain. So in RawAlign, we add a step to that. What you can see over here is alignment, where we take the output from chaining and we reevaluate it. But at this point, we do it at a much finer granularity. So before, we had a relatively coarse-grained, fast evaluation methodology where we could take huge amounts of data, right? A reference genome on the orders of gigabytes, and read set that could also be equally large, perhaps even larger. And we use really fast and efficient data structures and algorithms. For example, these hash table lookups, as far as you can compare sequences, that's about as fast as you can potentially do it. The downside is it's really coarse-grained. So over here, for example, when you look at chaining together multiple anchors, you're really never looking at the contents of the sequence over there. Right? So anything that's between two anchors, it's ignored for the chain. The chain just kind of assumes, yeah, probably what's in between kind of matches. But it could be that what's the actual sequence contents in between make no sense at all to match together. And chaining would ignore that because it just checks the positions of the anchors. In alignment, you actually look at the sequence content. That makes it a much more expensive algorithm, but it makes it a fine-grained analysis, and that makes it also much more accurate. You now use essentially all the information you have about your two sequences at that location, and then that gives you a much better way to decide in the end if that candidate should map or not. Yeah, please.
So I thought that like the sequencer just continuously reads the entire molecule that it has. Mm-hmm. But like in, for example, the first graph, that's like not the case. Like how does it decide where does it do the read?
Where it does the read in the molecule?
Yes.
Okay, so how you actually run these sequencers is that you take a biological sample from somewhere, you add some chemistry, you put it into a centrifuge, so you kind of really extract the DNA molecules. But during all that process, you have essentially, we call them library preparation protocols. So depending on the exact recipe you follow for preparing whatever you put into the sequencer, you, for example, break up the long DNA molecule into shorter pieces. Not so short as short reads, but perhaps 10,000 bases. Or you try to have a protocol that keeps them in very long pieces. Whatever you do, at the end of the day, you just have kind of a volume of sample in there, and the sequencing machine just randomly pulls one every now and then.
Yeah, sorry, I think I've confused gamers and reads.
I see. Okay, that's an algorithmic. Gamers are just an algorithmic choice, right?
Right. Yeah. So each of these dots on the graph is like how many bases long, right?
Each dot over there is one seated. I think in RawHash it's the concatenation of how many. I think 4 kmer. Well, no, it's 8 kmers. And then actually how many bases that really spans, it depends. I guess 12-ish. No, that order of magnitude at least. So essentially what one hit here says is that you have something on the order of roughly 12 nucleotides that matches really well between the read and the reference. Yeah.
And okay, so I think I understand like what's happening on the left. And so how does it get the alignment on the graph? Like how does it know what's happening in between the reads?
Well, so on the y-axis, it's a reference, right? X-axis, sorry. So over here, you looked at to extract these seeds in the first place. You looked at the contents of the read, right? You picked, okay, let's take this cable and look it up in hash. Here you ignore the contents of the read. And here you just look back at that floating point array. And you know where in that floating point array your seed, for example, came from. Then you can check, okay, so we have, I don't know, nucleotides, well, or raw signals over there that somehow match or don't match the reference. But maybe let me go here to the next slide. How you actually compare these arrays then is with a dynamic programming algorithm. Similar to chaining, right? These dynamic programming algorithms will a 1 or 2-dimensional or n-dimensional table. Here it's 2 dimensions of integers or floating points. And they usually start in the maybe top left. They fill out the table according to the cell-wise update rules that are represented by the arrows. And once the entire table is filled towards the bottom right, you get your solution bottom right. So in base code analysis, we, for example, use something like Needleman-Wunsch or Smith-Waterman, which are exactly these type of algorithms. So what you would have somewhere in your program, if you implement this, is this kind of update rule that the dynamic programming entry ij is somehow determined by this formula. Right? It can either, it's the minimum of the top left, or the left. And for the top left, you additionally compare the nucleotide sequences at that respective read and reference position. So somehow, whether they match or don't match affects the score that you compute. So far, so good? Right. And yeah, this is the point where I mentioned we can essentially get inspired by base code aligners or base code mappers. We can do very, very similar things for raw signals. Right? Raw signals, whether they're actually whether they're actually represented as integers or floating points, it doesn't even matter too much. The point is it's numeric values of a relatively large range. We're working here not with quantized values, by the way. We only quantize the values for the sake of seeding and chaining. Here we will go back to actually using the raw original values because we want to analyze it in as fine granularity as possible. But we have a very similar structure, right? We have a dynamic programming table, and these update rules are somehow the minimum score of those 3 neighbors. The only thing that's different here is how we count similarity or dissimilarity of the sequence. So instead of just comparing the nucleotides, are they equal or non-equal, we just get the absolute difference between the numeric signal values in the reference and the read. The idea here is that the numeric signal value can have noise. It can be almost the same. And that should still be considered as matching reasonably well, even if it's not a perfect match. So using an equal sign would not be the right choice. On the other hand, if something is significantly different in the numeric value, then it's really a mismatch, and it should be considered as a huge difference. That's why we have kind of an arithmetic operation there instead of just a comparative one. Was that your question about how you compare them?
I think it was something else that I already.
Okay, very good. So there are some challenges in integrating alignment to mapping. If there weren't, I guess everyone would already have done it. Certainly in base code read mappers, alignment has existed for a long time. We know it works in principle. We want to have alignment. But what's particularly challenging here, and especially for raw signals, is that alignment algorithms are called frequently. So you have to imagine that for every chain, there are very, very many chains. For every chain that you identify it, you later have to reevaluate it with alignment if you decide to use alignment, right? And it turns out there's really many chains, and in RawHash, for example, it just generates the chains and it picks the best one, the one of the best score. If you want to reanalyze all of these with alignment, you have to do it for all of them. And each time you call one of them with alignment, it's extremely expensive. So why is expensive? Recall that this is how this somehow looks, this alignment matrix over there. Calculating this is slow. So the reason here is that these dynamic programming tables, if you do it naively, it grows with the square of the sequence length, roughly. Right? So you have essentially a table, the dynamic programming table that I showed. It essentially has an entry at every y coordinate and it has an entry at every x coordinate. So here you already would have like a 200 by 200 table or more, and that's about 40,000 entries. A dynamic programming table of 40,000 entries that you need to calculate for arithmetic operations. That's expensive even on modern fast CPUs. Compare that to chaining, which just has to like compare 1, 2, 3, 4, 5, 6 anchors for the same read. So the fact is, efficiently integrating alignment requires some algorithmic innovation and, let's say, also some engineering to implement those innovations efficiently. So we developed some techniques here where we use pre-alignment filtering, also chaining. Then we develop an early termination strategy where we realize sometimes we just don't need to do alignment actually. We actually don't do alignment for all chains, only the ones that look promising. We have anchor-guided alignment, where somehow the information we get from chaining reduces the number of entries we need to calculate. And then we can do some banding windowing, which essentially means we only follow the diagonal in the dynamic programming table instead of the full table. And finally, we can also use hardware techniques, of course. In this case, we just used vectorization. In fact, we let the compiler just vectorize the code. We had to massage the code a bit to let the compiler do its job, but in the end, it can just always do its best version for that CPU, and it does so automatically. So all of those implementation details are in the paper, but we can now look at some evaluation and see if we really got any benefits out of those algorithmic changes. So, like in RawHash, we compare to uncalled and SIGMAP. We have slightly different CPU than in RawHash, but I guess those are all details. And of course, we use many threads to evaluate this. Then we have multiple use cases: read mapping, relative abundance estimation, contamination analysis. So those are essentially the same data sets that are also used in RawHash. We have 5 key evaluation metrics. So the memory footprint. Of course, alignment doesn't really change the memory footprint, but as an overall tool, as an overall method, we'd still like to have that. Then the throughput and the analysis latency, those are significantly affected by, first of all, the computational overhead, but on the other hand, the additional accuracy that we get out of alignment. The sequencing latency, that's mainly impacted by the accuracy that we get, and the accuracy itself is, of course, its own metric, right? That you already had, and we plot this really nicely as spider charts here. So a larger area is better. Yeah, one of each of these peak points corresponds to one metric. The spider chart grows larger the better you do in all of these metrics. Okay, so here's RawAlign in blue, and the baseline tools including RawHash. And as you can see, as a initial observation here, that RawAlign is really the only tool that does well in all metrics. And yes, you can always pick out individual metrics where another tool does better. For example, we can look at OnCall, which has, I don't know, really low memory footprint. But it turns out it doesn't really have any reasonable accuracy. It doesn't have any reasonable analysis latency, and it doesn't have any good throughput. I mean, essentially reverse throughput. So what really matters is that you have a tool that does all of them reasonably. Right? And as it turns out, RawAlign does really well in all of those. That turns out generalizes well across all data sets. So across all data sets that you can see here, RawAlign always has the largest area of any of the tools, largest area in the spider chart. So yeah, if you look at, okay, one particularly funny one is relative abundance over here. Here we have uncalled, for example, which has extremely high throughput, extremely low memory footprint, and very low analysis latency, but does so without any tangible accuracy whatsoever. So I would actually say, although it does well really high throughput over there, because it's so extreme in the metric that it satisfies well, and so extremely bad in the metrics that it doesn't satisfy, uncalled here would almost be a bit of a useless tool in that data set. In contrast, you know, RawAlign, it has, let's say, the key metrics are covered over there. By the way, throughput, it looks bad over there, but it only looks bad compared to uncalled, which has insanely high throughput while doing nothing. Right. Especially on these difficult data sets, we observe that RawAlign is the only tool that has really high accuracy. In particular, RawHash, which was the only other tool that can deal with these large data sets somewhat reasonably, we significantly improved accuracy. Right? We have doubled the F1 score. Almost. Right. Of course, we report all of these results as raw numbers, but I think we don't need to do this to ourselves here. Another interesting bit here is on the relative abundance results. So far, we didn't compare to base call trees members. So far, we only compared to other raw signal tools. In the relative abundance case, we have the unique opportunity that we can kind of end-to-end evaluate this. So relative abundance calculation means you have a sample that's mixed from multiple species, and for every read, you want to determine to which species does it belong. And in the end, hopefully you estimate correctly how your sample was composed, right? For example, oh, there were 50% of human cells in there and 20% of COVID cells and so on. Since we know how we composed our samples, we have a ground truth, and then now we can compare both the base called approach with Minimap2 and the raw signal tools below to the ground truth. So there you can see the distance, lower is better, right? That's the distance to the ground truth. Instead of the art base calling by base called baseline, wow. It's not at zero distance. It has like a distance of 0.05, which is low, but it's not zero. Now the other raw signal tools are kind of a huge distance if you compare it to Minimap2. So they hugely struggle with correctly estimating the number of reads because they have accuracy issues, especially on this relative abundance data set. But it turns out that RawAlign is the first of these raw signal tools that does kind of reasonable. Clearly, it still works than the base call pipeline. We're still less accurate, but that's okay. We at least did so without needing a huge deep neural network to do base calling, and these tools are also much less engineered at this point than Minimap is. That's been out for years. So let's at least say as a claim that RawAlign approaches the accuracy of state-of-the-art base calling-based analysis, and that's promising enough in and of itself. As these tools develop, hopefully we can get closer and closer to that, and eventually maybe reach that accuracy or even surpass it. Yeah, there's of course much more analysis in the paper, but let's start going into the discussion soon. So we saw that prior tools kind of fail to scale to large reference databases in one metric or another, not necessarily performance, sometimes also in accuracy. Our goal was to satisfy all of our key metrics for a wide range of reference database sizes. And to this end, we proposed RawAlign, first tool that combines these seeding, filtering, and alignment steps for raw nanopore signals. And we observed that it really does well for large reference databases. It works actually for all kinds of reference database sizes, and especially compared to RawHash, it barely loses throughput, but significantly improving the accuracy. Right. Any questions? Otherwise, we can quickly go into an analysis. No questions. Very good. You understood everything. Great. So I'm analyzing my own paper here, but I tried to hold myself back with strength. First of all, RawAlign improves on state-of-the-art numbers, right? Accuracy, runtime, decision latency. That's really the key goal at the end of the day. It's also really nice that RawAlign is tested on it's built upon well-tested algorithmic principles. I mean, we didn't have to do any voodoo magic, I guess, right? We added alignment that conceptually makes sense because we know it works from base called read mappers that also use alignment for high accuracy. So we kind of can sleep well knowing that we're not doing something weird. But at the same time, there are some challenges in there to actually implementing those well-tested algorithmic principles, and RawAlign solves those using, say, more algorithmic techniques, careful implementation. We use a bit of hardware acceleration to do it. So we kind of do our best at engineering this as reasonable as we can based on well-known algorithmics without getting too fancy and getting very clear results that works. This is a proof of concept that we should in the future always do alignment for raw signal numbers. All right, there are some weaknesses, unfortunately. There still certainly remains a significant accuracy and performance headroom to be explored. We don't have 100% of one score yet, nowhere near, in fact. So especially for the large databases that are more difficult to be accurate on, we get as high as maybe 75% of one score. That's not where we want to be, of course. Realistically, we'd like to have 95, 99%. Second part is kind of tangential to this entire raw signal analysis direction. The downstream analysis is kind of missing for now. So yes, we can, for example, use this for real-time analysis and then do read until decisions, which is all not fine and dandy. But when we do traditional base called read mapping, usually what we actually want to do is later decide, does a patient have perhaps an increased risk of cancer? Or is there, I don't know, we want to perhaps understand if they have any genetic diseases. Those downstream analysis tools that run after read mapping, they're for now at least missing for raw signal mappers. That means we are yet to show that this is also useful in this way. And that's, of course, a huge gap. In that way, for now, no matter how accurate we are, we actually cannot compete with base code remappers because the, let's say, the infrastructure, the software infrastructure doesn't exist. Okay. I note down a single discussion point. And I note down some answers, but maybe you can quickly think about it. How can we potentially make RawAlign faster than it is now? Because performance is still an issue. What can we potentially do? Where can we start to make it faster? I don't expect a concrete algorithmic idea. Rather, what are the places you could try to tweak things? Lisa, do you have ideas? No? Okay, no genomics for you. Do you have any ideas? It's a difficult question. You know, I asked myself this question for how can we make it faster, and it was really easy to write some things down. And then I asked myself, okay, how can you make it more accurate? And then I actually, yeah, for making it faster, there's maybe some halfway obvious things you could potentially do. First of all, you could try to do filtering before chaining. Right? So potentially you can develop algorithms to, even before you start analyzing with your, well, I said chaining is an efficient algorithm, but it's still, because of the amount of data that goes through, it's still actually significant amount of the runtime of the entire tool. So if you could somehow reduce your data before you start chaining, or for example, just removing the worst 20% of seeds, you know, you say, okay, look, these 20% of seeds, they don't have any other seed hits around them within 100 characters. If you could just get, with such a strategy, you could just get rid of maybe 20% of your seeds, that's significant improvement. Later, after chaining, perhaps you could add more filters. And so some algorithm that's cheaper than full alignment, doesn't have this n square runtime, hopefully maybe even linear runtime, and it doesn't need to tell us too much. It just needs to eliminate the absolute worst of candidates. So we reduce the data again. Of course, you could always tweak the actual alignment algorithm. Maybe you don't do dynamic time warping, right? You don't do this dynamic programming table. Maybe there's another technique to efficiently compare signals that actually works in this context. There are many ways to compare a pair of floating point arrays or integer arrays, right? The question is, does it apply to this type of data that you typically see in those arrays? For example, you could estimate the Jakar similarity of these, the reference at the read. And then you would have to work out if that's actually now sufficiently accurate still. But my point is, alignment isn't the only way to do it. Perhaps in the end, of course, I mean, a large part of our group works in hardware acceleration. So maybe Lisa, well, she's not working on hardware acceleration. Maybe she can come up with some other smart ideas to use computer architecture to just do the same computation still, just do it faster, more efficiently with engineering techniques, essentially. All right. Yeah, just call the entire field of hardware acceleration engineering. That's not fair. Yeah, that's all I have to say. Please let me know if you have any questions, and otherwise, yeah. Yes.
Can you maybe go back again to the three graphs?
Yes, you can. Somewhere. Fine. So
we have the data that we get from the sequencer is an array which spans like basically the entire x-axis.
Well, this is an excerpt, I guess, right? The array on the x-axis would go from 0 up to, well, it depends on your read length, right? It could be 10,000, it could be 50,000. The very longest reads, it's a stochastic process. The very longest we've ever seen is 2 million, I think.
Okay, yeah. That makes sense. So how do you get the positions for the seeding?
How you obtain them?
Yeah.
Okay, so the reference is always an excerpt, right? In reality, this 300, 400, 500, it's relative to that offset up there, 4.5976. That's actually a huge y-axis. And along that y-axis, that's where you build your hash table in React.
Yeah, sorry, no, but like how do we select the positions on the x-axis for which we look up?
Okay, very good. You essentially take every, so there are some techniques in RawHash that subsample, but as a first starting point, you would take every position. So we take the raw signal sample at every location in the read, you query the hash table every time, and then it's the job of chaining, of actually making sense of all that data. Here, rather than having, I took an excerpt which is actually the correct mapping location. But if you look at the entire thing that you get out, this is really a huge plot, right? It's millions of lines in length, and it's more of a point cloud.
So for example, for position 150, like reposition 250, there is some point, but it's somewhere else.
Almost definitely, and probably that point is on many places on the y-axis.
Yeah.
This is rather a point cloud, and chaining finds actually some location in the point cloud that fits together well.
Okay, I think it's
yeah. And actually, out of chaining, you will probably get multiple locations in the point cloud that fit together well. You will probably get this kind of nice diagonal thing of multiple seeds. You'll find it many times, and all of them then you pass on to alignment. That then tells you which one is really the best, instead of just guessing. Very good. All right. Then thanks for your attention. Anyone on Zoom? Thanks for Nisa's attention. And yeah, I guess, is there any closing words? No? We can end it here. Are you stopping the live stream?